Right.
Definitely.
For that?
Yeah.
Okay. That's what I'm about to go over. Tell you what. Mine, gosh, I don't even know if we can get everybody around
I
here.
can send the video.
Okay.
Oh, can you send the one you just had?
Yeah, I can send them up.
What an age we live in. All right.
Send a group message, just send it to everybody.
Yeah.
Right.
Yeah, how do you?
That works.
So
just have
then
to go in
send it
your
to me and say,
settings
oh
and
my
turn it
God,
on.
is that what I sound like?
Yeah.
All right, so here we go with a cross section through
When he
the
fills
spinal
this out.
cord.
Yeah, yeah.
I don't know.
Here we've got a vertebra surrounding, so the vertebral foramen is surrounding the spinal cord. So this is a cross section through the spinal cord right here. And so wrapping it, shrink wrapping the spinal cord is pia mater. There are three meninges, the outermost dura mater, then the arachnoid mater, and the innermost pia mater. So the pia mater directly shrink wraps both the spinal cord and the brain. So this is definitely pia mater here. And then we have the spinal cord. The pia mater, anteriorly, you know, the body of the vertebra is the front, spinous process is the rear. The pia mater anteriorly folds into a deep gash in the spinal cord called the anterior median fissure. So this gash in here is the fissure. And the pia mater actually folds into and lines the anterior median fissure. It stays around the outside posteriorly. So you've got this groove called the posterior median sulcus that is not lined by pia mater. It stays completely on the outside. The hole in the center of the spinal cord is the central canal of the spinal cord. Then we've got gray matter and white matter. All of this, coming across all of that, is gray matter. This is white, this is white, this is white matter right there. And the key difference between gray matter and white matter is this. Gray matter lacks myelin, white matter is loaded with myelin. The white matter looks white because of all of the myelin. Myelin doesn't stain. Myelin is very, very resistant and remains very pale. So, gray matter, the parts of neurons without myelin, white matter, neurons with myelin. So what parts of a neuron, or what part of a neuron will be myelinated, or can be myelinated? Just the axon. So you've got millions of axons, myelinated axons making up the white matter here. And then you've got axons that aren't myelinated, plus all the other parts of the neuron that are never myelinated. Dendrites, cell bodies, teledendria, axon terminals are never myelinated. So they would compose the gray matter. All right, so we can divide gray, by the way, another way to tell if we were to remove, which I can't do, but if we were to remove this vertebra, you should still be able to tell front from back. Anterior median fissure, posterior median sulcus, plus the gray matter extends all the way to the posterior edge. Extends all the way to the posterior edge, but it does not extend to the anterior edge. There's white matter in front with the gray matter anteriorly. So these are ways to get oriented on the spinal cord. So the gray matter is divided into horns, gray horns. So this would be the left dorsal or posterior gray horn, the left lateral gray horn, and this little tip right there is the left anterior or ventral gray horn. So right dorsal or posterior gray horn, right lateral gray horn, and there would be the right anterior or ventral gray horn. All right. Then the white matter is divided into columns. So we have the left and the right anterior or ventral white column, the left and the right lateral white column, the left and the right dorsal or posterior white column. So the white columns are predominantly made up of ascending myelinated axons and descending myelinated axons. Okay, now we come to the outside of the pia mater. This great big space all the way around is called the subarachnoid, out here, the subarachnoid space out here. The subarachnoid space is filled with cerebrospinal fluid, CSF. Then we come to, remember, so we have pia mater, the deepest of the three meninges. Then we come out to arachnoid mater. Then finally we come out to dura mater. So you've got this big gap, the subarachnoid space between the pia mater and the arachnoid mater. So the arachnoid mater and the dura mater fuse together. So I'm running the chopstick over the dura mater, and my fingertip is touching the arachnoid mater. Okay, so the arachnoid mater is just inside, dura mater just on the outside, right there. So you see that black groove separating them? They really do join together. There's a virtual space between them that does not contain any CSF. But that virtual space, this very, very narrow virtual space between the inside arachnoid and the outside dura mater, that's called the subdural space. So you're going from from pia mater to subarachnoid space, to arachnoid mater, to subdural space, to the dura mater, to the epidural space. Outside the dura mater, where all this yellow adipose tissue is, right there, that is epidural space filled with adipose tissue and veins here. All right. What else? So, yeah, so we've got here, we've got this yellow bulge. Guys have another one of those models? Can you guys see this okay? Can you see this all right? All right, so to make sure you guys can play along at home, so to speak. This yellow bulge is a ganglion right there. Here's its partner with dura mater covering it. So remember, dura mater's on the outside, and that continues over all of this. So dura mater comes out. This is all dura mater out here. This is all dura mater here, here, here. So it's been peeled away to reveal the ganglion here on the right. Its partner ganglion on the left is still covered by dura mater. So what kind of ganglia are these? These are dorsal root ganglia. These contain the cell bodies of unipolar afferent neurons, rather, not axons, neurons. They're sensory, in other words. Then that means that they're going to be conducting in the afferent direction to the central nervous system. So the cell bodies give way to the axons. So this yellow structure right here is a right dorsal root. So right dorsal root ganglion, where the cell bodies are, right dorsal root, where the axons are. These are all afferent axons conducting to the back of the spinal cord. Then the dorsal root splits up into rootlets here. So these single strands here, these rootlet single strands, these contain many fewer axons. They're smaller axon bundles. So we're splitting up all the afferent axons into smaller rootlets. They're contained within smaller rootlets. So the dorsal rootlets, right side, left side, come into the rear of the spinal cord. So the dorsal rootlets then meet the dorsal gray horns within the spinal cord. So conduction to the spinal cord is into the rear, is into the posterior spinal cord. What about going out? What about efferent? What about leaving or exiting? Well, in the front, we've got these other rootlets. So you guys can swing this around for you guys here. These are the left ventral rootlets and the right ventral rootlets. They contain small numbers of efferent axons. So they're coming out. Conduction of action potentials will be coming out, and those ventral rootlets combine all of their axons to form a ventral root. So this is a right ventral root there, right ventral root. We don't show it on the other side. So to give you a sense of what's going on in terms of conduction pathways, you would have a sensory impulse coming in to the rear of the spinal cord, and then a motor impulse going out the front. A sensory impulse coming into the rear of the spinal cord, motor impulse going out the front. What I didn't mention was this thing connecting across the midline. That's called a gray commissure, right there, gray commissure. A commissure is an axon bundle that crosses the midline. And that's what we mean by commissure, an axon bundle that crosses the midline. So the central canal goes right through the gray commissure, you see there. What they're not showing you, because they screwed up, is that immediately anterior to the gray commissure, there should be a white commissure. So the gray commissure is a bundle of unmyelinated axons crossing the midline. Immediately anterior to the gray commissure, there should be a white commissure, a bundle of myelinated axons crossing the midline. but they've got the pia mater and the anterior median fissure going in too far. They eliminate any white commissure on here. Shame on them. But here's the point. You can have ipsilateral conduction. Impulse coming in posteriorly and leaving anteriorly on the same side. Entering posteriorly, leaving anteriorly on the same side. Or you can have contralateral conduction where you cross the midline by way of one or the other commissure. So you could have conduction coming in the rear, crossing over, going out the front on the opposite side. Coming in the rear, crossing over, going out the front on the opposite side. So commissures allow the impulse to cross from one side of the body to the other. Okay? So they're pretty important, in other words. Any questions so far? Anything at all comes to mind? All right, we're not done. There's more on this model. Let's see. So, where are the nerves? Where are the spinal nerves? Turns out, remember I said when I was going over this thing, the nerve begins lateral to its ganglion. The nerve is the axon bundle, the ganglion is the cell body bundle. The nerve begins immediately lateral to its associated ganglion. So, the associated ganglion, we'll use this one, our right dorsal root ganglion. If you go lateral to it, you come to here. So this is the nerve, right there, guys. There's the ganglion, there's the nerve, covered by dura mater, right there. There's the ganglion, there's the nerve. Turn this back around for you guys. There's the ganglion, there's the nerve, both covered by dura mater. It's short. It's also, this is the nerve. This is the nerve. But look what it does. It almost immediately splits into two. It splits into a dorsal ramus and a ventral ramus. A smaller dorsal ramus, a larger ventral ramus. Ramus is just Latin for branch. So it branches into a dorsal and a ventral ramus here. And it's worth noting, I mentioned all spinal nerves are mixed. They all contain some afferent and some efferent axons. The same with the rami. So the dorsal and ventral ramus both contain efferent and afferent axons. So even the rami are mixed. So you have both incoming and outgoing impulses to the back, incoming and outgoing impulses to the front here. Now, so the ventral rami on this particular model have what's called a communicating ramus with these structures here. These structures, see this little bulge, left side, right side? This is another ganglion pair. So this is a left, this is a right sympathetic chain ganglion. So I'm that around for you guys. Ventral ramus, communicating ramus, sympathetic chain ganglion. Ventral ramus, communicating ramus, sympathetic chain ganglion. So if the dorsal root ganglia contain the cell bodies of afferent neurons, the sympathetic chain ganglia contain the cell bodies of efferent neurons, motor neurons. So totally different, in other words. And by the way, sympathetic is a clue about location. The sympathetic neurons are all located, you might want to write this down or record it. Sympathetic neurons are all located from T1 to L2. So you only find sympathetic axons in 14 pairs of the 31 spinal nerves, from T1 to L2 inclusive. There are no sympathetic axons in any of the cervical spinal nerves. There are no sympathetic axons in L3, 4, 5, or any of the 5 sacrals or the coccygeal. So all sympathetic axons, autonomic sympathetic axons, are bundled together with other axons, they're not the only ones, in spinal nerve pairs T1 through L2 inclusive. So 14 out of 31 pairs are where you're going to find sympathetic axon. So my point is, these are sympathetic chain ganglia, and they are, then we must be from T1, must be in the T1 to L2 region. Would you agree with that logic? Okay? Can anybody tell me what kind of a vertebra that is? It has these vertebral arteries running through a left and a right transverse foramen. Which vertebrae have transverse foramina? Which 7 vertebrae have transverse foramina? These cervicals. So this is a cervical vertebra. What the hell is it doing around thoracic spinal cord? I demand an explanation. It's the wrong vertebra for this model. Does not belong on this model. There should be a thoracic vertebra because most of the sympathetic nervous system, it connects to the thoracic portion of the spinal cord. From T1 to T12, then a little bit of lumbar, L1 and L2. So there's almost certainly a cross-section through thoracic spinal cord. There's another clue. See that lateral gray horn? That lateral gray horn? They contain the beginnings of sympathetic pathways, T1 to L2. So these two bulges are found, the lateral gray horns are only found from T1 down to L2. So that's it. So again, that's where we are. More further proof that some dummy put the wrong vertebra on this model. Okay? I must write a stern letter one of these days. Any questions about this model? I think that covers everything on here. Are your brains full yet?
Yes.
Getting there? Okay.